All right, hello everybody. It's my pleasure to introduce our speaker and also hi to people who are following us on YouTube. It's my pleasure to introduce our speaker today, James Wordsworth from Newcastle, UK, the University of Newcastle. He's at the Department of Aging and Health that doesn't exist anymore apparently, organizationally. But anyway, so he's from Newcastle, and James is actually a very interesting person, a very critical and open-minded person. He has some very good original ideas, and this is why I thought it would be very nice to hear what he thinks about the theory of aging, which is a burgeoning field today, and he has some interesting insights into that he's going to talk about today. James actually graduated from London. He did his undergrads there at the Imperial College, and then he moved to Newcastle, where he is now, and then he also did some trips to Nice, as I learned. He did some postdoc there, and then when he was fed up with the palm trees, then he went back to Newcastle to do a postdoc and at the department that I just mentioned. And then so we are very interested to hear about your work, and thank you for accepting our invitation.
Oh, thank you very much for having me, thank you for the invitation. So I'm James, and I'm going to talk to you today about a unified model of aging. So what do we currently know about aging then? Well, we're always introducing our papers by saying something like aging is a complex multifactorial process, or aging is a rope of events. Aging is a complex process. But to me, this is like a map that just says you are here. It's basically a glorified shrug. It's like beginning every paper by saying we actually have no idea what we're talking about. So, this is obviously not where we thought we'd be right about now. We've ended up in sort of this very strange and quite scary place. So the first thing I wanted to ask really is, how did we get here? So I'm going to split the talk into three parts. The first one asking that. The second one asking, is there a better way to look at aging? And the third part being a theory which I think could explain much of what we know about aging. So part one, so how did we get here? So in the beginning, aging was simple. It was wear and tear. It was a thermodynamic requirement. So just as the cart ages and dies with time, so does the horse. But whilst entropy is always increasing, so whilst this is true of the cart, it's not actually true of the horse. There is no thermodynamic imperative for aging. We're open systems. We can take energy from the environment to maintain ourselves indefinitely, which meant that aging was a biological problem, not a physical law. So it was subject to evolution. But then how could something that is so clearly negative evolve? Did it offer something positive? Perhaps at the group level. So early ideas were there would be some benefit to the group, perhaps with increased genetic diversity for the aging organisms, which would allow them to outcompete the negatively senescent ones. But individual selection is now sort of mainly thought to trump group selection, and negatively senescent cheetahs would arise within our aging population, and they would outcompete the self-sacrificing aging organisms. Tom Kirkwood argued very persuasively against programmed aging, and the idea of an altruistic program has been widely dismissed. Though if you want an interesting counter opinion, I would recommend Cracking the Aging Code by Josh Mittledorf. So perhaps then aging was beyond evolution to correct. Perhaps this detrimental thing happened because it occurred after organisms were already expected to be dead from extrinsic causes, meaning that it would just be too difficult for evolution to select for anti-aging traits. However, we now know that aging is widely observed in the environment, and organisms experience big fitness costs as a result of aging. Aging can also be highly malleable, highly plastic. You can have, for example, in ants or termites, you can have workers that will die very quickly and queens that can live for 30, 40 years, which suggests that aging is not beyond the capacity of selection. So what if aging provides an advantage to the individual, perhaps earlier in life? So the benefit that they gain doesn't have to be quite as large as the obvious cost. So this was antagonistic pleiotropy, George Williams' theory. However, whilst we found some of these antagonistically pleiotropic genes, none of these could explain sort of aging except as a combination of troublesome processes. However, the vast majority of genes that we found that have big effect in aging all fit within this sort of same pathway or a network, which I'm going to call the aging module, which is largely consistent across species, and these mutations can obviously have massive effect. So whilst there might be other genes which are having like smaller effects, it does look like much of aging could be explained by this single process. So could we then have a single theory to explain the bulk of aging? Along came disposable soma theory and free radical theory, and this was like the perfect mix of evolutionary and mechanistic explanation. Aging was thought to evolve by accumulated molecular damage because we were simply just not investing so much in maintaining ourselves, so we could put that energy into reproduction and growth, and that would allow us to produce more offspring and outcompete organisms that were investing more in maintenance and managing to be negatively senescent. So this was kind of like the perfect antagonistic pleiotropy. However, there are several arguments against disposable soma theory. What I think is that the main problem with it is it ignores homeostasis. For example, if we were to invest less in our antioxidant defenses, that doesn't mean we can just plow that energy into having more children. Instead, we would have to increase investment in repair. And if we don't do that, we end up with more misfolded proteins, and we have to invest more in correcting those misfolded proteins. And this actually requires much more energy than we would save by not investing in our earlier defenses. Equally, if we don't invest in these, we end up with either, if we get bigger problems, we have to kill the cells off. And if we don't kill the cells off, we're left with incorrectly functioning cells. And again, a damaged cell is significantly less energy efficient than a functional cell. So either we have to replace the cells, which again requires more energy, or our other cells are then just going to have to make up the work of our damaged cells. So again, we're not actually saving any energy. So if disposable soma theory is incorrect, then we actually we actually lack any evolutionary underpinning for the idea that aging is a result of accumulating molecular damage. What about the mechanistic details? So we have the mitochondrial free radical theory. It started off with some really promising correlations between oxidative damage and lifespan, but after that it was sort of this spree of failures really, from the failure of Gates to a bunch of papers openly questioning whether the theory had any right to continue. So do reactive oxygen species affect maximum lifespan? The answer was really no. Telomere shortening, again the answer was no, there didn't really seem to be any effect on maximum lifespan. Senescent cells, okay, we could get median lifespan, but again no, there was no maximum lifespan benefit. Meanwhile, the empiricists were at work and they were looking for, well, they found all of these genes, all of these behaviors, variants and drugs, which were all having massive effects on our lifespan, and they all seem to be affecting this aging module. We also have parabiosis, and we have these Yamanaka factors, which can completely rejuvenate cells by removing the epigenetic marks, which is what allows all organisms, including Dolly, to be born at age 0. So Jean-Pedro de Magahay said, well, perhaps aging isn't a hardware design flaw after all. Perhaps it's a software design flaw. Perhaps it's these epigenetic marks that are causing aging rather than actual damage to the DNA. However, he still suggested that this would be a random process. It would be random epi-mutations, as he calls them, accumulating over time. Yeah, I don't think it does need to be random. We have these first, second, third, and fourth generation clocks now, all with a remarkable ability to track our chronological and biological age. So could these potentially have some specific function? So where does that leave us? we have this sort of good intuition that aging must involve some kind of damage. We have a seemingly unconnected aging module and some plots, which appear to measure age pretty accurately. So how do we fit all of this together? At the moment, the main way we seem to be doing this is through inflammation. So we have this kind of system where damage is thought to lead to inflammation, and then we have some kind of positive feedback through insulin resistance, which is then activating all this stuff, and then that feeds back into more inflammation, which leads to age-related disease. So this is about the best systems-level understanding that we have of aging at the moment. But of course, it makes no sense. Like, the entire aging module is limited to this positive feedback back into inflammation. So where does that leave us? Aging is a complex and multifactorial process. So we now appear to be in what is essentially a post-explanatory period of research. Everything, instead of is looking for the ultimate cause of aging, we're just linking it to these hallmarks, and we're trying to link these hallmarks to diseases, or we're just throwing drugs and, hoping something sticks. And that's because we no longer believe that aging is a simple process. But then we still have this aging module, which seems to explain most of aging. So could aging be a simple process after all? Or perhaps slightly more complicated than we initially thought? So part 2, is there a better way to look at aging? So even if aging is a complex multifactorial process, could we perhaps elucidate which processes are having which effects or which outcomes? And I thought a good place to start with this would be the Gompertz law, which is that basically we experience this exponential decline with aging. And it made sense to see sort of which diseases were following this same pattern. And this is true of all the major age-related diseases: cardiovascular diseases, sarcopenia, dementia, Alzheimer's. But what is not so true for is cancer. This is, I should add, a logarithmic scale, so this is much more exponential than it looks. Like we still have the childhood cancers, and these are in many ways almost anti-age-related diseases. We have these really weird profiles over here. And even the conventional cancers, they still have this drop off. So perhaps cancer could be sort of a separate process to the rest. Some more evidence for that. So we have these genome-wide association studies, and the one that they always pull out is apolipoprotein E, something associated with lifespan. The ApoE4 allele is associated with the least long lifespan, whereas ApoE2 is always associated with the longest lifespans. And ApoE4 is obviously associated with all of these diseases, but the allele most associated with sort of earlier cancer is in fact ApoE2, suggesting it's kind of the opposite, inverse relationship. The same thing is true for FOXO3A, which is the sort of the next one we always pull out. It's mainly associated with cardiovascular disease and Alzheimer's. It has very little relationship to cancer. And then there are things like low-density lipoprotein levels, which increase with age, and they're like a really solid biomarker for aging. Again, whilst they correlate massively with these diseases, they correlate negatively with cancer. So could these perhaps be sort of one aging process, and this other group bit another one? And there is, in fact, one disease that really does correlate very well with cancer, and that's fibrosis. Basically, wherever you find, so this is idiopathic pulmonary fibrosis, which is the fibrosis of the lung. We also get it in the liver. And basically, wherever you find fibrosis, or wherever you find cancer, there's usually some evidence of fibrosis and vice versa. So fibrosis is essentially a wound healing response that's gone wrong. Normally, we have this relationship where fibroblasts and macrophages activate each other, but they're both still very dependent on the initial signal. And as long as that the initial signal dies in time, these cells will go back to sort of zero state. But if you have repetitive injury or prolonged injury, what you get is potentially sort of enough stimulus that you don't need the inflammatory stimulus anymore. These cells can just activate each other over and over. and you end up with essentially an almost unstoppable positive feedback loop. Like the only real cure for idiopathic pulmonary fibrosis is a lung transplant. So why do people think that cancer and aging or cancer and the other age-related disease are the same? Well, we have somatic mutation theory to explain the advent of cancer, and we have all these damage-related theories which are thought to describe the rest of aging. But whilst this has sort of been largely proven true for cancer, at least, fundamentally at the basic level, that is obviously much less true for aging. The evidence just hasn't really come through for that. So could these perhaps be different processes, process 1 and process 2, I'm going to call them for now. So enter diabetes. So type 2 diabetes is a disease of the diet. In hunter-gatherers, it probably didn't even really exist. For a long time, it was basically limited to the rich, the poor mainly escaping it by not getting enough food. But now it's basically everywhere. Diabetes is obviously completely inconsistent with the idea of aging as we understand it. It's more like plane crashes or viruses. The ultimate cause is very clearly extrinsic. Yeah, weirdly, metabolic syndrome is so massively interrelated with these age-related diseases that it almost does seem like the same process. And even stranger, it's also highly related to cancer and fibrosis. So, are we perhaps looking at another process, which I'm just for now going to call process 3, but I'm immediately going to change that to what I'm going to call extrinsic aging, which is essentially aging that's very easily modifiable by extrinsic factors. So extrinsic aging actually gives us a lot of power. It can explain why genetics seems to have so little effect on our mortality. It can also explain why extrinsic behaviors seem to have so much effect. Yet at the same time, it allows for this other underlying process, which is almost unalterable by any of these behaviors. Like even our top athletes are still showing massive decline over time. Just put this into sort of a little order. So potentially what we've got here is initial cause, followed by what I'm going to call intrinsic aging, which is largely unalterable by these things, could potentially be slowed, but there's almost no evidence that you can really reverse this. There's nothing in nature that really seems to be getting younger so much that you couldn't explain by this extrinsic aging process, which is massively modifiable by things like stress, your diet, exercise, and then this is leading to metabolic syndrome and age-related disease. So we have semmelporous organisms which die after mating. So these deaths often look more like deaths of stress and exhaustion. But during mating, the male antechinus, which probably you guys are more familiar with than me, but this guy essentially whips himself into such a frenzy that he gets plaques in his brain, which are equivalent to Alzheimer's disease. Yet, if you keep these guys away from other males and you give them enough food and water, they're fine again after that, and they can live for another 6 months, and they're perfectly healthy. They can't mate again, but so could potentially this seminal parity be just massively accelerated extrinsic aging? So now we've named both of these processes. We just need to work out what they are. So to find out, we should probably start again at the beginning. So that's part 3. This is going to be sort of an attempt at a theory which can explain most of what we understand about aging. I'm going to start with the intrinsic aging. So the current model is we get DNA damage, and that basically causes an increase in noise, cell loss, basal inflammation. But what if there was another type of mutation that's actually impacting selection within our tissues? So what if potentially, for example, we had a mutation that affected a cell's basal metabolic rate? For example, a cell could grow faster, it could replicate faster, it would just be doing everything faster than the other cells. And again, oppositely, we would have cells that are growing slower, replicating slower, and have a slower metabolic rate. And just for the rest of this talk, anything red is going to be associated with being slow, and blue will be fast, mainly. So obviously the slower cells are going to be sort of naturally outcompeted by your wild type cells, whilst the faster cells will have a natural selective advantage within the tissue. They're going to outcompete them, and they're going to gradually spread throughout the tissue. And just to reiterate, these are not cancer cells, they're normal cells, just with an elevated metabolic rate. Tissue homeostasis is largely maintained, no tumours are formed, but basically if there's a gap, these cells are more likely to be the ones to fill it. So there's actually plenty of evidence this is happening. So this could be something that causes aging, right? It's a change over time in a single direction. But is this actually what aging looks like? Well, I mean, what we kind of predict from this is you get sort of the tissues, they're getting faster and faster, so just gradually depleting all our resources until you get death. And I think we can all agree that that's not really what aging looks like. But there are diseases that look like this. And these diseases are probably cancer and fibrosis. So they are overactivity disorders, uncontrolled growth, positive feedback. And this is process 2, which I'm now going to call celeresis, meaning the generation of speed. So what if this process 2 or celeresis is just the spread of these fast cells? And we already know that the predictors of this celeresis are sort of adverse to these other predictors of intrinsic aging. So so could intrinsic aging essentially have evolved to stop this process of celeresis? And could aging therefore be a mutant defense mechanism? So we know that we do have defenses against these mutants. Largely we look at this in terms of sort of an autonomous process where we have something like the activation of an oncogene like RAS causing hyperproliferation, lots of DNA damage. We activate a tumor suppressor and the cell kills itself or it becomes senescence. But there are still smaller changes in growth that don't cause these massive effects. Like they're just having more minor effects on cell growth. They're not causing this hyperproliferation and massive damage. The DNA remains largely intact with a maybe slightly elevated mutation rate. So it's really difficult for this cell to know whether it's a mutant or if you've just had a big lunch, you've got lots of insulin, IGF floating around the system, and both you and all the cells around you are all moving at a slightly faster rate than you were before you had the lunch. So these cells cannot just autonomously remove themselves. There's actually a study done in 2017 by Nelson and Marsall which suggested that the spread of these uncooperative mutants, as they called them, would inevitably lead to multicellular aging. But, of course, this isn't the end of it. Just because this cell itself doesn't know that there's a problem, that doesn't mean that the cells around it couldn't know. If cells can compare their growth rate to a neighbour, then you could have very minor differences in growth, and these cells could go, you're moving at a different rate to me, I think that's a problem, and I'm going to remove you. So we modelled this, where we had cells that could get faster or slower, and we basically we allowed the cells that were next to sort of a similar metabolic rate cell to protect each other, while cells that were different would try and remove the cell with a different growth rate. So we gave the faster cells a bunch of different levels of growth advantage, and we increased or decreased the probability that like cells that are different would kill each other. And then we looked to see if basically a mutant would overtake or become the dominant cell type over the wild type cells. So initially we assumed that this process would be totally unselective. Any sort of difference in growth rate would be treated equally. And what we found was, when we did this, it didn't matter at all what the probability of killing the neighbor was. Like the fast cells would always become dominant, and they would usually be reasonably quick, which suggested that perhaps Nelson and Marsall were right. This was a sort of inevitable result of selection at the cellular level. However, we then decided to give these slower cells a survival advantage So basically, if a cell was moving faster, everything would be exactly the same. But if a cell was slower, it could then say, maybe you guys are the problem and not me. And it would have a survival protective mechanism. So we said that essentially, in comparison to the sort of unselective removal of the cells before, we called this selective destruction. And what we found when we did this was that everything completely changed. You now had this period where this bit in the middle where you'd essentially balanced the growth advantage with the survival advantage, and it allowed complete control of these uncooperative cells. So it suggested that Nelson and Miles were wrong. Aging is not inevitable. You can control natural selection at the tissue level. But what we decided next was that the cells don't actually even need to kill each other. It would be much more efficient and much better if the cells, if the slow cells, instead of killing the faster cells, they just induced an epigenetic change, which basically would slow the fast cells. And then we could start to explain why we're seeing things like these epigenetic clocks. And the best candidate we have for this is notch signaling, which I don't actually have time to go into. But so now we have these different states where we have loss of homeostasis with faster cells, or ceresis, and then we have this loss of homeostasis where the tissues are getting slower, or metabolic slowdown, and then we have this level where you've balanced everything and you have negligible senescence. So this appears to be the solution then. You just have to balance the growth advantage with the survival advantage. But there's a problem with the solution in that the growth advantage is not always constant. So if you have sort of the basal level, you have a sort of a distinct advantage of the faster cells here. Then you stimulate everything with insulin and IGF. Suddenly you have the faster cells are going to be much more responsive to this. So you have an increased difference in growth advantage. So it's possible that you have to choose between having a survival advantage, which is too high some of the time, or a growth advantage, which is too high some of the time. So you have to choose between hyperactivity or hyperactivity. And obviously, the advantage of hypoactivity or metabolic slowdown is that you're much more likely to be able to control these faster cells spread, but at the same time, you're causing these slower ones to spread, so you get metabolic slowdown. So it basically has reduced ceresis and thus reduced overactivity disorders. For example, if you get a tumor cell forming in this group of slow cells, well, that looks a bit like a bull in a china shop. These guys are like, whoa, you're going way too fast. Okay, so I'm going to get rid of you. Whereas if you have the same thing happening in these cells which are all moving a lot faster, then this tumor cell is suddenly somewhat less noticeable. You have this really sort of nice tumor microenvironment forming. So perhaps ceresis is sort of one of the things that's leading to carcinogenesis. So metabolic slowdown gives us these advantages that we're getting less cancer and fibrosis, but at the same time, the cost is intrinsic aging. And we published this in MechAgeDev in 2022. I think I made two boneheaded moves here. The first was calling it selective destruction, because it's not destruction, it is essentially, we're now arguing an epigenetic process. And the second was calling it damage independent, which is obviously not, because it's damage dependent. So the damage is still the initial cause here, both intrinsic aging and ceresis. So in the old model, we had lots of cells getting lots of different damage, and we had an accumulation of noise, basal inflammation. In the new or different model, you instead have a cell that gets damaged, and then that cell spreads damage by spreading itself, and that leads to this process of ceresis. Or you have a cell that gets slower, and this time it's not actually the damage that's spreading, but this cell is spreading its slowness epigenetically throughout the tissue, and that's causing intrinsic aging. So obviously this is not a new idea that aging has evolved as a cancer defense mechanism. There's been loads of really cool stuff done, argued most persuasively by Alex Wolff probably. But these ideas are still all dependent on this older paradigm of damage accumulation, and even talking from Alex Wolff, he fully accepts that this cannot explain aging in these post-mitotic organisms where they don't really get much cancer, or any cancer really, except in their mitotic tissues. And of course, you can't explain it in yeast. So Alex thinks this is not really a problem. Perhaps these guys age differently. Well, there's no competition happening in these post-mitotic tissues, obviously, but essentially then why do we have this aging module, which is in many ways even sometimes like more powerful in these organisms? So a key question, I think, is why would these organisms be post-mitotic in the first place? They're sacrificing so many benefits associated with cell division. You have lost regenerative capacity. You can't regulate sort of outputs by having changes in cell number. It really reduces your capacity for size. And it's not even the sort of the ancestral trait like before, like 550 million years ago. We had flatworms that would cut up into loads of pieces. They'd all form new worms. And then suddenly C. elegans went, whoa, that's enough of that. There's worms everywhere. And it became post-mitotic. So really the question is why would organisms do this? Like perhaps C. elegans' sort of mitotic ancestor like lacked a lot of the control mechanisms that we see today for stopping this runaway regeneration. Had massive or more problems with these cancers and fibrosis. So post-mitosis would be like a decent evolutionary strategy to avoid these problems of cell division. Of course, that's massively speculative. I'm not wedded to any of these ideas. But anyway, the point is that post-mitosis doesn't prevent all selection at the tissue level. Because even within your post-mitotic cells, you have organelles which are doing basically the same things. And you have your mitochondria, which are replicating independently of cell division, and they're doing mitochondrial dynamics are basically controlled at two main levels, replication and degradation, which is almost exactly the same as cells. So we could potentially be having, in these post-mitotic organs and organisms, selection at the mitochondrial level, which is basically having the same effect. In fact, selection within mitochondria is actually already sort of quite well sort of understood. Aubrey de Grey's survival of the slowest theory for mitochondria is just one example. Basically what this leaves us is we have DNA damage, and that's causing our cells, post-mitotic and mitotic, to be slowing down. And that intrinsic aging is designed to stop this sort of runaway cell process. But the result is this metabolic slowdown, and that is causing this extrinsic aging and age-related disease. But what is extrinsic aging? So that's the final part. So why is this happening? Because we have homeostasis. Essentially, all of our cells and organs are trying to keep all of this stuff as close to equilibrium as it possibly can. Our fuels for respiration are ATP, the main product of respiration, and what we use for the vast majority of our metabolism, all of our essential ions and elements, all the building blocks. All our cells are trying to keep these as close as they can to equilibrium, because if we don't do that, we die. And the more important the metabolite is, the faster we die. So previous sort of models of aging have largely ignored homeostasis. They've looked at aging as a sequence of vicious cycles between inflammation, damage, and senescence. But the body doesn't, obviously it does have these systems of positive feedback, but generally it tries as hard as possible to avoid positive feedback, and is largely built on systems of robust negative feedback. So what I'm suggesting here then is that basically as our tissues and cells are slowing down, they are using less ATP per minute, as pretty much by definition. That's what you do if you have less metabolism. You're using less of the unit of metabolism. So rates of ATP hydrolysis are decreasing, and that means in order to maintain our ATP homeostasis, we have to adapt. We have to restore this ATP homeostasis, and this kicks off this cascade which causes extrinsic aging. So if we want to keep our ATP levels constant with age, which most studies suggest that we do, when you're using less ATP, it means you have to produce less ATP. So this is in our mitotic tissues. In our post-mitotic tissues, we have the slowdown of the mitochondria, which I haven't really described very well, but talk to me later if you want. That's having the reciprocal effect, where your mitochondria are slowing, producing less ATP, so we therefore have to use less ATP. But what this both amounts to is that as our mitochondria are producing less ATP, that means they are burning less fuel, less sugar, and less fat. And that means, as they're using less, this is starting to build up in our bodies, in our cells. So what does a cell do when it's got too much of this stuff? Well, I mean, sugar's not so much of a problem, but most of our cells are not really equipped to have excess fat building up in them. So what do? They import less sugar and less fat, which means they take the insulin receptors along their membrane and they start degrading them, so that to the point where they're now at their new sort of metabolic equilibrium. And what does this result in? It results in insulin resistance. So this is why we become more insulin resistant as we age. And of course, as our cells are taking in less of this stuff, it's now just accumulating in our blood, which of course we don't want. So what do we do with it? We put it in our fat storage cells, our adipocytes, which is why we gain weight as we age. Now, adipose homeostasis is actually pretty cool, I think. So the main way we control this is in adults at least, we have a set number of adipocytes. And when one adipocyte gets too full, it secretes a bunch of inflammatory cytokines, and that attracts macrophages pretty much dead on to the cell, and they kill it, and they take the fat, and they get rid of it. And that maintains really nice adipose homeostasis, except when you have a chronic change causing more and more fat to be stored, in which case you get more and more inflammation to attract more macrophages to kill more adipocytes. And at the same time, of course, you're going to have your metabolic slowdown of your immune cells, leading to an increased reliance on innate immunity. And both of these factors together are combining to cause chronic inflammation as we age. And eventually, even our adipose homeostasis is lost in our subcutaneous fat, where we try to store most of our fat. That's where it's healthiest for us. This supply becomes exhausted over time, and we start activating mechanisms to push all the fat to our viscera, all around our essential organs. And when we do that, of course, this is problematic, because we now have exactly the same inflammatory processes, except they're happening around our main essential organs, and they're causing highly destructive processes. But what this all leads to is essentially metabolic slowdown is inducing this metabolic syndrome. And actually, if you look at the criteria for metabolic syndrome, 4 out of 6 of those are immediately already described in the process I've just described: elevated triglycerides, serum glucose, abdominal obesity, high BMI. So this could be the product of metabolic slowdown. So others have basically described these age-related diseases as essentially metabolic syndrome in overdrive. Like some people have suggested that we rename Alzheimer's disease to type 3 diabetes. Benjamin Bickman is a big sort of believer that cardiovascular disease would largely be pretty rare if it wasn't for metabolic syndrome. So metabolic slowdown clearly leads to metabolic syndrome, but how does this lead to age-related disease? I'm just going to go through one quick example. So cardiovascular disease then is where we get sort of plaques building up in our arteries, and when these plaques break off, it results in stroke or heart attack, or in the lungs, pulmonary embolism, or it just causes general havoc basically wherever they end up. So the plaques basically block the blood and the nutrients and the oxygen from getting to our essential organs. You also get the blood vessels bursting, and that quite frequently results in death. So the main sort of hypothesis for why this happens is the cholesterol hypothesis. So essentially we get these fatty streaks in our arteries, where we get basically molecules of LDL building up, and then macrophages come along, they try and remove this. There's just too much, so they basically get stuck there, and they form these foam cells, and you basically get this ever enlarging fatty plaque. That's not actually so obviously this connects quite nicely with the idea of metabolic slowdown causing metabolic syndrome by the buildup of these fats and these triglycerides. There is another hypothesis, which again, I don't have time to go into at all. I actually find it more convincing, but I'm still working out how it fits. Anyway, so we modeled this in another paper, which is currently available on bioarchives. Hopefully it will be available soon. But essentially what this all means is that the risk and health span are all products of this extrinsic aging. And interventions which are addressing extrinsic aging are largely going to just result in this compression of late life mortality, a nice increase in health span, but they're never going to do anything for us in terms of actually massively increasing our lifespan. They will affect our median, but not our maximum. So intrinsic aging then, as obviously it's causing this sort of this exponential rise in mortality as our cells get slower and slower and slower. And that is obviously resulting, as it increases, it results in increase in risk. But if we look at extrinsic mortality, then it's just sort of at any given age, you have your sort of your best or your worst, based on what sort of all these extrinsic factors like stress and diet. At the beginning, extrinsic aging is not all that relevant, but later on, the effect is absolutely massive. So that's why, I think a lot of these interventions are proving so successful even at late ages. But obviously to achieve larger benefits in lifespan extension, we must look at intrinsic aging. So how do we do that? There are already interventions which affect our maximum lifespan. So calorie restriction. So one thing that's doing is obviously if you restrict the calories, you're removing all this extra these unnecessary fuels, you're removing all the burden on your adipocytes, you're preventing all this inflammation, and that's why CR improves our health span. But at the same time, what CR is doing is whilst you have sort of your cell sort of selection and competition going on, you have essentially like the faster cells. If you've got lots of sort of insulin and IGF bounding around, they're really running away with themselves, and they look very different to the slower cells. And that's obviously changed when you've got sort of very little going on. Everything's sort of slowed down, and your fast cells look much more similar to your slower cells. So you're reducing the rate of this selective destruction, and so that's potentially why CR could be affecting our maximum lifespan. And the same thing is obviously true of rapamycin. It's reducing our basal metabolic rate, and it's having massive, really sort of good effects on our maximum lifespan. But what rapamycin is not doing, that CR is doing, is it's not removing all this excess fuel. And that's why if you take rapamycin at the wrong time, you can induce glucose intolerance and diabetes, and you can have all these metabolic problems. So this is just from the model. So we just showed that, if you take it here, then you can just get increases in inflammation and fat levels, but at the same time, you're getting this benefit in the long term. What about exercise? So again, exercise is as our muscles increase in size and we're burning away all this stuff, we're having really potent effects on our extrinsic aging. So that's why exercise is really sort of, an excellent sort of source of metabolic health. But what exercise is absolutely not doing is it's making our fast cells look more similar to our slow cells, which is why exercise never really has any beneficial effects on maximum lifespan, I think. So there's also the Yamanaka factors. So if we have our fast cell that's been epigenetically slowed and you add all these like epigenetic removers, what you get is essentially a rejuvenated fast cell again. And that's why we can get this sort of amazing epigenetic rejuvenation of our cells. When we're trying to do this in worms, I actually don't really know this. This is just hearsay. But I've heard that essentially when you put this in worms, you just turn them to mush, because essentially what you're getting is a bunch of really rejuvenated worm stem cells. Don't hold me to that, but that's what I've heard. Right. So what about if instead of doing this entire sort of converting all our cells back to stem cells, we could look essentially at this process and the specific epigenetic marks which are causing our chronological aging? Could this be essentially how we could cure aging? But of course, the downside to that is if you do this in the wrong cell, you're going to get the cellulosis and lead to more cancer and fibrosis. So no one ever really asks why cancer evolves, to my knowledge. And partly I think that's because cancer is just thought to be another part of aging. However, if now we look at cancer as the product of cellulosis instead, then what we see is that cancer is essentially just natural selection occurring at the tissue level. It doesn't need to provide the individual with any advantage because it's just tissue level selection happening naturally. And this is sort of clearest in this canine transmissible venereal tumour, where the cancer cell has evolved in this dog and just become its own sort of new species of pathogen. So in conclusion, we have this process of cellulosis, which results from damage, and essentially intrinsic aging could be a mechanism to prevent that, and then that will lead to all these processes inducing metabolic syndrome through extrinsic aging and age-related diseases. But at the same time, what we're doing is we're producing this chronic basal inflammation state, which is basically, although our cells are getting slower, there's lots more inflammation to produce this sort of fibrotic, nice tumor microenvironment, which is why extrinsic aging also causes these diseases. So aging then could be a mixture of three processes: cellulosis, intrinsic aging, and extrinsic aging. So I'm not suggesting that anyone just buy this straight away. Obviously, there's a lot of very speculative stuff here. But what I would like to suggest is that, you know, it's, as a null hypothesis, something to sort of compare to. It's just like it's either this or this. So yeah, that's it. Thank you. Thank you. Yeah, thank you very much for listening. Big thanks to Adam for inviting me and for the university for having me. So I've had really good fun thus far talking with Adam. So yeah, if you want to email me, it's my email. I'm embarrassed to say this, but I'm also writing a book. It's not available yet, or will be for probably a long time, but yeah, hopefully one day. Okay, thank you.
Thank you very much. A lot of things to unpack here, and that's what I wanted to mention, because I already knew this, that James is working on his book, and I think I'm not the only one who's really looking forward to reading it. So yeah, I'm sure there are a lot of questions. So.
Oh, perhaps not.
Yeah, maybe I'm wrong.
I have a lot of questions.
Go ahead.
Hello, you were talking about calorie restriction with relation to aging. I'm not sure whether you're talking about fasting or maybe you were talking about lower caloric intake. I'm not sure. Could you clarify that, please?
Yeah, so actually, I think that's a really good question. So I think they have different effects. So I think if you lower your calorie intake, what you're going to get is you're going to get a much lower basal metabolic rate, and that will probably have sort of more beneficial lifespan effects in terms of reduced selective destruction and intrinsic aging. Whereas fasting, like, you're not going to get any sort of, like, if you sort of fast for 36 hours and then you're back to, you know, stuff in your face again, you're not going to have any sort of consistent lowering of your basal metabolic rate. But what you are doing is just having these really great periods where you're clearing all of the excess stuff from your body. So you're going to be really lowering your rate of extrinsic aging. So they're having two different effects.
Thank you.
Maybe a follow-up question, not a question, it could be a comment, that it's true that our real life experiences, what do we eat actually, whom do we interact with those psychological issues, is the one that could affect the aging and the one also would affect the metabolic activity in our bodies.
Yeah, so you mean like having good social interactions and reduced levels of stress. Mm-hmm. So yeah, so this all interconnects really nicely with sort of the whole idea of metabolic syndrome. So you have sort of the what we call the HPA axis, where you have essentially releasing of these stress hormones, and what these stress hormones do is they prepare your body for sort of fight or flight, and the way they do that is they constantly make sure you've got these fuels available, and what that means is you're drawing like fat out of your subcutaneous tissue, and it ends up sort of pushing it in your viscera. So I think that is partly why. So that is basically one of the big effects of cortisol, is it causes you to accumulate fat around your organs, as opposed to the healthy way of storing it. I think that is probably a big contributor to why we see these huge effects of really good social interactions and negative effects of sort of continuous stress and cortisol elevation.
Okay.
Yeah.
Okay.
One of the many questions?
Yeah.
Yes, so I was just missing what induced the metabolic slowdown, and did this metabolic slowdown is appear in the unicell organisms as well?
So, metabolic slowdown, so what I was proposing in the tissues is that it is a result of cell competition, and sort of, not so much competition as cell comparison. So you have your cells in your tissue, and they see something that looks dangerous, and then they attempt to slow it. It doesn't even have to be sort of looking at it as this cell looks dangerous. It's this cell's moving faster, and really we want this tissue to be sort of a uniform response, so it does everything nicely. So in unicellular organisms, obviously this is not a factor at all. But what you still have is you have your mitochondria within your yeast, for example, and they are competing with each other. If you have a mitochondrion which is really resistant to being degraded, then obviously you'd expect that to outcompete the rest of your mitochondria and would spread over time. So in our proliferative tissues and in yeast, like a really good way of getting rid of this is to have just one cell inherit all of the rubbish, which is why we don't see sort of the same sort of level of mitochondrial aging in our proliferative tissues as we do in our brain and our muscle. And yeast, obviously, they manage to get rid of all of the rubbish in one offspring, which just gets worse. Yeah. So feel free to disagree.
And other factors like, for example, cold or hypoxia, which can change metabolism in the cells. Is it how can affect aging?
Hypoxia, that's a big one. I don't know. I'm not really an expert on that. I'm very interested in looking into HIF as a sort of as a way that we sort of split glycolysis and and mitochondrial respiration. But I'm not going to try and answer that.
But I can see what normally is that incretin analogs can affect some. Is there any research that they can affect the aging process? I'm not going to answer that because I think I would be talking fluff at you. But I read some books by Jason Fung. I think he talks a lot about the different effects of sort of insulin stimulation from what sort of just general what you're eating as opposed to specific effects of sugars and stuff. And that he could give you a much better answer than anything I would say.
Yeah, thank you very much for this very interesting talk. If I understand well, your model of aging is based on the assumptions that slow cells can survive better.
Yes.
Why do you expect that? Because slowing down metabolism probably, at least for me, might mean that efficiency is also diminishing in this cell. So I would expect the other way around, that slow cells become less efficient, so can die.
So it's not like a natural selection for slow cells. What it is essentially a program where it's because the faster cells are obviously the ones that you would normally expect to just overtake and become the dominant tissue, or the dominant cell within your tissue. In order to prevent that, we have essentially this system where a cell will notice if something's moving faster and slow it down. So it's not sort of natural selection, it's a very artificial induced process
where you're deliberately giving the advantage to the slower cells.
But do you mean artificially induced process? This is induced by you, the creator of the model, or it's induced by higher level natural selection acting on the organism?
So what it is essentially an organism that has this system where you have essentially notch communication between two cells, an organism that has a system where instead of just sort of standard mutant removal, you have a system where it's mutant removal, but it's more likely that the slower cell removes the faster cell, that organism will survive better because it will get less cancer and fibrosis than an organism that simply allows
It's
the
a
faster.
result of the selection acting on the individuals.
So it's still individual level selection, but it's selection for individuals that have better tissue level selection. Yeah. So totally, yeah.
Thank you.
The back? Oh, the back?
Yeah.
Okay, we extend the lifespan of organism using the trade restriction or rapamycin. So
Using what, sorry?
Rapamycin or the trade restriction.
Yeah.
So what the intention of the theory of aging? Is it to maintain the productivity of the organism until they die, or just to live?
So the question is, what's the purpose?
Why we need to extend the life of organism unless they are productive?
So, I mean, the longer you live, the more offspring you can have. Is that a sufficient example? Yeah? I sense I've not answered your question.
Other than organisms like animals, in case of animal productivity, in case of birds, you can't say, we extend the life of their life, probably, but unless they become productive, what the intention of this theory of aging?
So, obviously you want to be, you'd want to be as reproductive and as healthy for as long as possible. And if you slow down the rates of intrinsic aging, particularly if you're more shorter lived, then that is going to be a significant benefit. And obviously, if you slow down your rates of extrinsic aging, you're healthier and you're fitter and you can outcompete other organisms. But I don't have any sort of specific thoughts on post-reproductive aging.
So thank you so much for the nice idea. You said that metabolic syndrome induces this, metabolic slowdown induces metabolic syndrome.
Mm-hmm.
So do you mean that this affects or increased aging or decreased aging?
So what I'm suggesting is that the metabolic slowdown is inducing metabolic syndrome, which is connecting all of these age-related diseases as sort of as one process. And if you can slow that, you're going to have very beneficial effects on your lifespan. But what you're not doing is you're not essentially reversing what we really call aging. You're just essentially making yourself much healthier and fitter for your current age.
So the follow-up question to what you once said, so this aging is evolutionary adapted by all organisms. So are we trying now to stop aging or to slow down aging? But why do we just age?
That's a big question.
Yeah. So why are we going against the charity rather to stop aging?
Well, so according to this idea, why we are aging is because because natural selection is occurring within your tissues for sort of cells that are inevitably going, if left to their own devices, what you're going to get is you're going to get cells which are going to outcompete your other cells. Just because you have a mutation and just by chance that cell within your tissue is fitter within the tissue. And that cell is going to overtake, outcompete, and that's going to cause problems for you. It's going to lead to increased risks of things like cancer and fibrosis. These are not the only examples. You'll end up, so for example, if you have a really fast cell in your pancreas, you can end up producing too much insulin. That'll actually kill you much faster than cancer or fibrosis. Like if you have these cells that are undergoing positive selection and you don't control them, you're gonna it can be really lethal for you. So because the best thing we can do to prevent this natural selection occurring is have essentially selection going the other way. So you counteract the natural advantage of these cells with a program which basically specifically removes these cells. But what that means is if you overactivate that program, you're going to push it the other way. Instead of everything getting faster and more active, you're just going to get slower and slower and slower until you have a whole different set of problems, which are essentially excess buildup of fuel, sugars and fats, sort of clogging up all of your systems and causing you to die in a whole different way than you would have died if you just let the fast cells take over. So it's essentially why we age is it's stopping us from dying earlier from other aging.
Why is it impossible to keep this process?
Well, that's a really good question. I don't think it is impossible, but I think, so we do have organisms that are obviously showing like really sort of good attempts at maintaining their homeostasis indefinitely. But I think there's always, like, there's a big risk of doing that, in that you're essentially allowing this natural selection more strength, because you're not attempting, so you're essentially alleviating the control mechanisms. So these sort of negatively senescent organisms might be more prone to sort of problems that you get. Like, obviously, in order to become negatively senescent, they're going to have much other better controls of these cells. But sort of everything else being equal, you'd expect them to have more risks of developing these sort of natural selection diseases like cancer and fibrosis.
Okay. So this is like a kind of thing that if you have to stop a plane at the edge of the cliff, you don't have to stop it earlier than...
Yeah, exactly. Yeah, exactly.
Yeah. So following up that question, I mean, very simple, but so is there more incidence of cancer and this kind of diseases, for example, in axolotls or this jellyfish that continuously regenerates, or?
That is a good question. I don't know the answer to that. I would love to know the answer to that. Like, I think it's unfair to compare these things like across species, because obviously every species is trying to adapt to its own intrinsic mortality, and it will have its own separate evolutions that allow it to do that. But no, I think that is really interesting, and I do want to look more into that. Yeah.
So just to answer your question, they actually have lower rates of
cancer. Oh,
yes, and for instance, like trichoplex, you can irradiate them with a huge dose of gamma irradiation. They have a huge DNA damage, and they are happy with that.
I mean, that is amazing.
So I think this is a really good summary for vertebrates, and maybe for some of the invertebrates as well, but like for flatworms, cnidarians, and trichoplex.
Yeah, I get that. Like, I do think that as organisms have become more complex, they become more at risk of these diseases. I don't really know how, but I think that is probably the relationship there. As you become more complex, you become more prone to.
Why?
That is the question. That's an important question. Yeah.
More chance to...
Yeah, I mean that's Peter's paradox, right? But actually, that is so. Peter's paradox is actually interesting. So with this model, as you get bigger, not with this. I mean, that's standard Peter's paradox. As you get bigger, you become more prone to cancer. But this model would suggest that as you get bigger, you are naturally less prone to aging because you have more cells. And it will just, more cells, and you have more time before they become slower and slower. So it should reduce your rate of metabolic slowdown as you get bigger.
All right.
Just one more. If I understand well, your model suggests that the somatic cells can notice that if the neighbor cells grow faster, and they can induce somehow to apoptosis of these
Or
cells?
slowing them epigenetically. Yeah.
Is there a proof of this? That somatic
Yeah.
cells can do this, because usually immune cells the serve.
Yeah, so I mean that's a great. So there's really good evidence for this in cancer, which is the only place we've looked. So there is essentially Notch is massively involved in the suppression of cancer cells, and people have shown like you mutate Notch, you get much more cancer, and it's not just mutating Notch in the cancer cells. If you mutate Notch in the cells around the cancer cells, then you get more cancer. So there is really strong evidence that these cells are controlling each other.
So not only immune cells, but
it's not just immune cells, no. Immune cells, obviously, the more advanced your immune system gets, the more we are probably relying on that. But yeah.
I think a follow-up of this, but I don't want to go into that detail because we're short of time, but maybe we can discuss later. But how about controlling locally, like you said, that by neighboring cells, and what about systemic regulation? And also I think that would be very interesting to look at proliferative tissues versus tissues where they don't divide as much, like, I don't know, heart muscle or brain or something, whether those cells are more sensitive to like this weirdos who start growing faster, or I don't know. But we can talk about this later. So we're going to go for lunch, and I think that the sad conclusion of this talk that above a certain age, those who join us for lunch are not recommended to actually eat, but just to look if you care about your health. But whatever, it's up to you. Thank you so much again, and then we can continue the discussion during lunch.